Translation is such an important piece of biology since you are who you are, your looks and all your traits, because of the proteins that you have. Translation is the process that makes those proteins, so I can't express how important this is and just how many questions on your test are going to come from transcription and translation. So we're going to do a quick review of it in this video and talk about it a little bit more. Sometimes just hearing it another time really does help. So if we just review the steps and how this works, first of all, mRNA has to get made first. So we're assuming at this point transcription has already taken place. So the mRNA that was made in transcription is going to be connecting to a ribosome. The ribosome is going to be reading those 3 mRNA nucleotides. It reads 3 at a time, and those 3 letters on mRNA are called a codon. The tRNA, which is transfer RNA, is going to bring the proper amino acid, the correct amino acid, and it is going to bond to the messenger RNA strand. At that point, the tRNA is going to give up the amino acid that it has been holding on to. It's going to give that up to the ribosome, because the ribosome's job is to bond those amino acids together, which ultimately make the protein. Proteins are made from those amino acids. And those steps of reading the mRNA, the transfer RNA bringing in the the transfer RNA bringing the correct amino acid, all of those steps are going to continue until the ribosome reads one of the 3 stop codons. Now in this picture, okay, we're going to go through some visuals because that does help some people. You have this broke apart into codon number 1. We notice AUG, that's kind of our start codon, that's methionine. Codon 2 and 3 and 4, and they are grouped in 3s. So 3 letters equal a codon. This large structure in the back here, this is the ribosome, and the ribosome's job is to be reading these 3-letter codes. So it's reading AUG. This is a transfer RNA, and the transfer RNA has the anti-codon on it, so the U and the A and the C that match the codon. And it also is carrying an amino acid, and they've abbreviated the names of the amino acids. But you notice that the next transfer RNA, the correct anti-code, C with G, G with C, another C with G, that's going to come in. It's carrying an amino acid, and what happens? The first transfer RNA leaves, and the 2 amino acids are now bonded together. And that process is going to continue. So when the next transfer RNA comes in, notice it's a different code, so it's a different anti-code. This transfer RNA will leave, and those 2 amino acids will come over, and now we have 3 amino acids, and that process continues and continues until you reach eventually codon number 7, which would be a stop codon. This is just another picture, and this one shows transcription. So right away I see the nucleus, and so I know that this single strand that is connecting and doing the opposite codes, and this is the double strand, there's your DNA, this is your mRNA, this is transcription right here. You are going to need to, on the test, be able to look at a picture and know this is transcription, this is translation. I know that this is translation out here because it's in the cytoplasm, and translation takes place in the cytoplasm. So translation, but here's my ribosome, here's my mRNA. I know that these weird looking things, transfer RNAs look different in lots of different pictures, but this is the transfer RNA because there's my anti-codon, here's my codon on my messenger. I have these amino acids which are being brought and connected to your transfer RNA so that they can get delivered to the ribosome. So you have all these things happening in these pictures. Now we're going to go through examples. We're going to go ahead and start with the DNA. We're going to do transcription, and then we're also going to do translation. So again, transcription takes place in the nucleus, and here is the DNA that you need to start with. I know it's DNA even if this wasn't labeled because there are T's in it. T's are not found in RNA. Now at this point you need to pause the video and you need to do the RNA that it's asking you for in the notes. And then you want to restart the video so you can check that you're right. So give the video a pause, make sure that you move everything into RNA, and then start it back up. The mRNA that you should have, okay, this should be what is on your paper. Hopefully you are practicing it, because I guarantee you will have to do this on the test. The mRNA, you can see all those letters there. They're a perfect complement to the DNA above it. And now we're going to move on to translation. So this strand of mRNA is going to exit the nucleus, and it is going to go out and bond to a ribosome. So if we take a look at the mRNA from transcription, this same mRNA is going to come down here. Exactly the same. It's leaving the nucleus, it's going out to the ribosome. And now you want to take that mRNA, notice I kind of grouped them in threes so it's easier to see. You want to get out your amino acid chart, and you want to look each one of these codons up, and you want to fill out here in your notes the amino acid that goes with that codon. So again, you're going to stop the video, you're going to look up your 3-letter codes, fill out your amino acids, and then start it back up and make sure that you did it correctly. If you're starting this back up, then your amino acids should be methionine, proline, aspartic acid, tyrosine, and then your last one really isn't an amino acid because it's a stop code. Now, there is a spot for you to do the anti-codon. Remember, the anti-codon comes from the original codon. So this AUG, what would be its anti-codon? 3 letters, the opposite of AUG. What are the 3 letters that are the opposite of CCU? And that's what you want to do for the anti-codon. So again, pause the video, try the letters out, because again, you're going to have to do this on the test, so you need to practice it, and then start the video back up. If you're starting the video back up, you should have the following letters. Now notice that this last codon, UAA, is stop. So when there's a stop, there is no amino acid, so there is no transfer RNA brought to the mRNA strand, so there is no anti-codon for the last set of 3. This is just one more picture. I think it's a little clearer than the previous one. Again, you have transcription. You even have it listed in here. Transcription, the DNA unraveling to form this single strand of mRNA. So here's your nice strand of mRNA that leaves the nucleus, and out in the cytoplasm you have this ribosome that's reading it. You can see the 3 letters here, the transfer RNA, the amino acids that are on the transfer RNA. We have all of the pieces. You have to be able to, from a picture, tell what process is transcription, what part is translation.